Um, so let's get started just by looking at the one. There we go. For statistics. All right, tough outermost meninge obviously is the Dura Mater. As opposed to the arachnoid, which is kind of the middle one that has a subarachnoid space where the CSF is flowing around, and the Pia Mater, which is the very delicate one right on the neural tissue. This hopefully, if make sure you are clear about the whole flow of CSF. It's being produced by the choroid plexuses, and then ultimately going, in the ventricles, out the lateral and median apertures into the subarachnoid space, from the subarachnoid space through the arachnoid villi into the dural sinuses, and finally from the dural sinuses draining into the venous circulation via the jugular vein. Embryonic hindbrain. Most people got, that it gets you the medulla oblongata, pons. A few people actually wrote pons as ponds with a D, like the thing a duck would be floating around in. So make sure it's pons, which comes from a root for bridge. So it's not related to like a duck pond. In the cerebellum. Some people, actually more than one, so a few people, I think this is where it's important to make sure you reread the question, because this question, which says embryonic hindbrain develops into which structures of the adult brain, they answered forebrain, midbrain, hindbrain. So there was obviously something off there in terms of like understanding what the question's actually meaning. So make sure, you're always kind of going back, reread the question, that you didn't just like thought, oh, hindbrain. I remember hindbrain's related to these other things. Go back and make sure you reread that you answered it. Choose all true regarding ganglia, neuron cell bodies in the PNS. That is correct. They're definitely not glia or the CNS. And again, back to myelin. We have oligodendrocytes in the CNS, Schwann cells in the PNS. So that is that.
Professor, we didn't get into too many details about satellite cells. Is that something you're going to talk about more, or do we not need to worry about that?
No, we don't really know much about them even. We know that they are kind of cushioning and supporting, probably providing metabolic support to the neurons in the ganglia. But at least as far as I know, people kind of, leave it there and pay more attention to kind of the sexier cells in the nervous system. There probably are people who are specialists in satellite cells, but they don't get a lot of love out in the literature. All right, so let us continue with the nervous system. We kind of laid out the big picture of how the brain is in your skull and is being kind of taken care of. What we're going to do now is put a lot of energy into just what are the different parts of the brain doing. And so let me share. So the brain, so here's the brain, like, and it's in your skull here. Here's you. So a few things I should mention. First is when people think of the brain, sometimes they kind of make this analogy of it's like a computer, which in some ways, on a very, very superficial level, you could say there are some similarities, like you have the inputs, right? Your senses are kind of coming in, kind of like your mouse and your keyboard and stuff like that. And then there's the processing, like the CPU is figuring out what to do. And then there's these outputs, like either you're moving or doing this. So in some level, you can think of some of these very superficial ways of how the brain or the nervous system is similar, like a computer, but there's also some really major differences that are worth kind of thinking about. One is how it manages to do really complicated processing. Like a computer is doing things really kind of super fast, but in kind of a linear fashion. Like a computer CPU, like central processing unit, can literally perform billions and billions of floating point math operations per second. So when you're, in modern computers, some of like the really fancy CPUs are like have 4 or 8 cores going at the same time even. But in general, computers are able to do amazing things because they can work really freaking fast. Your neurons are really slow, right? If you want to talk from one neuron to the next, you've got to like release neurotransmitter, let it diffuse across a synapse, let it bind some receptors on the postsynaptic membrane, which then goes through some whole big convoluted second messenger pathway to finally open up ion channels, which create a voltage change on the second neuron. So there's delays around synaptic transmission. Like you want to send something down a axon, even the fastest axons are just going like 100 meters. We're talking about milliseconds, whereas thousandths of a second, whereas in computers we're talking about like billionths of seconds for things to happen. So part of the way, but your brain can do crazy fancy calculations and control, like creating a system that will control a body and make it go where you want. Like it's only been recently. I mean, you've probably seen on YouTube now, they've got these robots that can kind of walk around or jump or dance and stuff like that. That's literally like in the last 5 or 10 years, computers are finally so fast that they can do the calculations to kind of walk and not fall on your face. Whereas your brain, people have been doing that since the beginning of humans, been able to run around or climb a tree. So we talk often about like massively parallel computation. There is a lot of different parts of the kind of computations in the problem that are all kind of being taken apart and distributed and processed in parallel and kind of converging on a solution. So even though the actual hardware, so to speak, of the brain is super pokey, like compared to a computer, it's literally like millions of times slower than the little flipping of bits in a computer, but you can still do super complicated processing because of this kind of massively parallel type of computations that it does. So other things that we'll talk about that are kind of different between like the way a computer works and the way your brain works. We're going to see just this kind of input, what you see is actually, it doesn't just kind of come in and get processed. Kind of what you see is actually influenced by like your state of attention. I'll talk about this more in a few moments even. like what you're paying attention to affects the allocation of your visual cortex in your brain to different parts of the visual field. So again, I'm just going through this to kind of give you a sense of, yes, there are analogies between your brain and computers, but it's also different. So blah, blah, blah. Other things, I'm going to be telling you all sorts of stuff about, oh, this part of the brain does this, or this part of the brain is doing that. Like, what are the tools that we use to actually figure out this story about what the brain is doing and how it's doing it and what different parts of the brain are doing what? I think it's worth having some sense of that before we dig in. So it's not just like, I guess I'll believe you. It's like, how do we actually figure this stuff out? I'm going to say this part of the brain is responsible for production of language. This part of the brain is processing visual information. This part of the brain is doing this. This part of the brain is weighing ethical versus pragmatic content of decisions.
Like fMRIs.
So one thing is fMRI. This is actually pretty modern. This is the stuff I've been using at Berkeley, looking at these, subjects who've been, taking MDMA with their therapy and trying to get a sense of, do their brains process information differently after the therapy than before? This stands for functional magnetic resonance imaging. Functional. So MRI it's an imaging technique. I'm not going to get into the details of it now. You can use it to get anatomical scans of things. People get MRIs of their knees and stuff. It's better at imaging soft tissue as opposed to like X-rays, which are good at imaging bones and stuff like that. So you can use it just for doing anatomical imaging, but this functional MRI works because it turns out that the signal that you get from oxygenated hemoglobin versus deoxygenated hemoglobin is different. So you can actually tell where blood flow is going and where blood has just delivered oxygen, what parts of the tissue are being more hungry, more metabolically active. And you can do this non-invasively. You can do this with somebody who's just a normal subject, any random person. You don't have to, worry about, I don't have to actually drill a hole in their skull and stick an electrode in their neurons to actually get a sense of what is the neural activity as they're looking at an image or thinking about an elephant or whatever. So this gives you this real-time information about neural activity in a healthy, aware person. So it's very powerful. It has a lot of problems too, though. It's very low resolution. Like you can see, we call them voxels. The parts of the brain that we can see are usually about, a few millimeters cube. And so we get the sum total of all the activity in some, a pretty large chunk of brain that's actually probably, thousands and thousands and thousands of neurons all working together. So you're not getting very fine detail about brain activity, but at least you are getting information. And there's lots of other, it's very bad time resolution. You can only see things changing on the order of seconds rather than, so that it's really cool. It didn't even exist really when I was doing my graduate work, at least not at the level that we can use it now. And now it's a really powerful way to kind of explore what is the brain doing. What are other tools?
Surgically, like post-stroke, like when they're getting a surgery to kind of try and repair the area to see if it still works.
So what we call lesion studies. So lesion studies, this goes back a long time. This goes back hundreds and hundreds of years or even more. But lesions are just kind of tissue damage. So if there is tissue damage, and it could be from a stroke, could be from traumatic brain injury, for whatever reason, you see part of the brain is gone, and then you look, what kind of deficits are there? How is this person different from a normal brain? And that gives you some sense of what was that part of the brain responsible for or involved with, right? That's how, like, a few hundred years ago already, they were, a couple hundred years, I should say, they already figuring out what parts of the brain are involved with language. They'd have someone with a stroke who lost the ability to speak, and then they could do an autopsy eventually after they died and see what part of the brain had actually been damaged. Or, you know, anyone who's been in psychology has probably heard about Phineas Gage, who got like a metal rod blasted through his head, you know, and took out parts of his brain, but he survived. But, you know, his personality changed to give you a sense of what the function of the parts of the brain that were damaged were doing. So lesion studies are important. What other ways do we try to understand the brain?
PET scans?
There's PET scans. So PET, positron emission tomography, this is a little more specialized. This is where you can actually use, you have to create specialized radioactive molecules, but that will like bind to your neurotransmitter receptors. So you can actually kind of watch where these different molecules are going, where they're binding in the brain. You can see them. It's not as common because you need to actually have some lab making these specialized radioactive markers that you can then use as part of the process. Whereas fMRI is something you just put them in you can actually just use a scanner that would be used for normal kind of diagnosis and stuff. This is much more specialized. Yeah, this is not for phyto. This means positron emission tomography. What else?
An EEG?
Yeah, EEGs are important. Electroencephalogram. So this is the electrical activity just measured off your scalp that's due to your brain, right? Because your brain is here in your head, and it's doing all sorts of electrical activities. But if you put some electrodes just on your scalp here and measure, you'll be able to actually have some signal here that you can measure on the scalp that is being generated by all the electrical activity of the neurons in your brain. So this is totally non-invasive. And it's very high resolution in terms of timing. You can get very good timing resolution as opposed to like things like fMRI and things. But the problem here is you have very low spatial resolution, right? You only know that there is some electrical activity here that's somehow related to the activity of all the brain. It really doesn't tell you what specific part of the brain is generating that activity there, but it's still very useful. In fact, we're going to be looking at this maybe later today for sure. By Thursday, it's one of the main ways we try to like understand sleep in a clinical setting. Is like there's very typical EEGs, electroencephalogram patterns that you'll see when somebody's awake, when they're asleep, when there are different phases of sleep, or if something's wrong with their sleep, for instance. So EEG, in fact, one of the clinical signs of being dead is like you don't have an EEG. So even if you're in a coma, there is an EEG. It's very slow, but there's something there. If you're like kind of dead, there's like no electrical activity discernible anymore there. So that's good. EEG is very common and important way to understand. What are other ways?
CT scan.
See, so CT is more is people do, that's more of an anatomical thing. So it's useful for looking at the structure of everything, absolutely. So CT, and then fMRI, again, like I said, MRI is used also for anatomical stuff, particularly because it's better at discerning soft tissue. In terms of functional stuff, there's one other thing I'll mention. Transcranial magnetic stimulation. So this is actually used quite a bit in research. So the idea here is your brain is inside your skull. So again, if you've been in physics, you know that electricity and magnetism are just two sides of the same coin. If you take a wire and move it through a magnetic field, it induces an electric current in the wire. So what happens here is we want to, you take this kind of, these coils and you put right near the skull. So this is non-invasive, but then you have these very directed magnetic fields that you kind of generate and collapse. And what happens is it induces electrical currents in the brain tissue right underneath it. So you end up getting electrical stimulation of the brain tissue right under the skull where you have this machine, which is kind of generating this magnetic field. And so you can actually get people to twitch. You can do all sorts of stuff. You can, and in fact, there's a way this is also even used to create what are called virtual lesions, where if you pulse it in quick succession, you can actually have that part of the brain kind of stop responding for the next 5 or 10 minutes. So you can see like what happens if you kind of inactivate parts of the brain. Supposedly it's safe. Like I've never volunteered for one of these. I know people who volunteered for them and said it actually feels kind of weird and cool, but it seems kind of weird. It's like, oh yeah, that part of your brain doesn't work anymore for 10 minutes, but then it's all good. All right. But so that's another tool people actually use that is useful, where you can actually, and then another one is, in fact, another way that people mapped out a lot of the somatotopy on the, we're going to talk about somatotopy on the post and precentral gyruses for your somatosensory cortex, your motor cortex, and how it connects to different parts of the body. You know, when people have surgery, particularly for epilepsy, they often actually have to take off the skull and do brain surgery. So they'll cut off part of the skull. Oops. So, and then, so here's the brain, and now the skull is open, and people can stick electrodes right in there and just measure things directly from the brain. And the brain has no pain receptors in it, so there's no weirdness in terms of like, ouch, you're poking my brain. So you know, people don't actually stick electrodes as much as often they'll like put an electrode on top. There's actually these labs, this one lab at UCSF that will lay whole arrays of electrodes across the brain, So they get lots of readouts. And again, these are people who have brain surgery for different reasons and then agree to be part of a study. And it's like, you know, okay, my brain is going to be exposed anyway, and is sure measure from it. And I'll do some little task while you see what my brain is doing. So that's another thing. You know, I should maybe mention while we're here, epilepsy. Again, a lot of times the reason people are having brain surgery is because of epilepsy. And so it's not seems like a good a place as any to just mention what epilepsy is. You know, these are, you know, we call like seizure disorders. So these happen because something goes wrong with this, you know, that idea of, you know, the threshold when an action potential is getting fired off, right? Normally you have, you know, your neurons, and we talked about, you know, when the hillock reaches some threshold, then we set an action potential, which goes down and goes to the next part of the circuit. So if the system is working properly, there's all this activity coming in here. And if it reaches some critical, you know, threshold, then it sends an action potential block. So if the system is working properly, this neuron only fires off if it's appropriate to the amount of stimulation that it's gotten and that the circuit is all working properly. In seizure disorders, you get these kind of hyperactive areas where it's kind of like a little electrical storm that gets started in the brain. So you know, it's kind of like a little electrical storm. You know, here's your brain. There's some little area we call like kind of the focus that just starts going off, gets hyper-excitable and starts firing off for no reason. It shouldn't be doing it, but it's gone over threshold kind of in an errant way, an aberrant way. But as soon as that happens, it's then going to start exciting the other things around it. So then that little electrical storm is going to propagate out and continue, and just, you know, worst case can actually take over the entire brain. We have what are called grand mal seizures. If that electrical storm actually takes over the entire brain, that's like those, like, where every, you know, because obviously all your muscles are also getting, you know, motor cortex is going, everything. So grand mal seizures are kind of everywhere, but they don't, you know, seizures can be more focused. You can have seizures that are just like in your temporal lobe and you just hear things. If it's like kind of a little electrical storm in your auditory areas. You can have absent seizures. We call petit mal. Here, you wouldn't even know anything is happening unless you like say, like you realize that the person has just checked out. It's like, hello there, anybody home? And nobody's home. So it's just like for a minute, you always think of, I always used to think of seizures or epilepsy as this kind of, you know, more grand mal thing, but you can have seizures where just somebody just, you know, checks out and they are unresponsive, they don't seem to be home, and then all of a sudden they come back. These are more common in children, and they often resolve just, you know, as the brain continues to kind of mature and wire up. Often these can show up in children and then just be, you know, be gone by the time people hit adulthood. But the thing that is pretty nasty about these is this idea of kindling. And this is this concept that the more that a part of the brain has experienced seizures, the more likely it is to have additional seizures in the future. It kind of, there is a kind of a damage that happens. And so if people are having seizures, you really want to stop it, because it'll just keep getting worse if you just leave it on its own in general. A lot of times we'll just use drugs. Use drugs to try to keep the thresholds down, keep the electrical activity less excitable. But if you are not controlling the seizures with drugs, then they will go in and surgically try to take out that area that is kind of that focus that's causing the kind of the root of the seizures. So that's why you end up getting people with their skull off and people going in and trying to get rid of that. Because actually I was really sad. It was like a couple of years ago, I had a student who had a pretty bad seizure, history of seizures. And it affected her memory to the place where she had to drop the class. She tried so hard to just learn this stuff, and she was smart. She worked really hard, and it's just her memories just kept evaporating. So you really want to try to control this if you can.
What about seizures caused by brain tumors?
So that would be kind of different, and that probably would resolve more once you kind of get rid of the tumor. But I think that's just obviously if you're causing some kind of trauma on the neurons that are getting kind of pushed on by that, I could imagine that's affecting their behavior. That I'm not as familiar with, though. So epilepsy, you should definitely know what it is. And yeah, oh my God, I can tell you, I'll tell you this just because it's horrifying and kind of weird. When they were trying to originally come up with medications for seizure medications, what medications would be useful for someone with epilepsy, they literally would have this model where they would put electrodes on the eyes of mice and take a voltage source and just crank it higher and higher and higher and see at what point did this mouse go into seizures. And then they would give it these different drugs, and the drugs that let you crank it to a higher voltage before the mouse went into seizures, that would be like a candidate for an anti-seizure medication. So just to give you a sense, like the history of, like when people say like not tested on animals, sometimes that's like a really good thing, because some of the animal testing is really horrifying. Like, I don't know, that this one, just for whatever reason, just always like, oh, that poor little dude. Anyway, so let us continue now. So we've kind of laid out this idea. There's the brain. We know quite a bit about it. There's a lot of stuff we don't know, which we'll talk about as we continue on. I mean, things that you really want to know, like, consciousness, that's still, it's more of a respectable thing to study now. It used to be not even that respectable to study. Now it's actually respectable to try to understand what are like neural correlates of consciousness and things like that. But we're still so in the infancy of, like, we can talk about how does the brain extract information from a visual scene or something, but like, how does that actually become your experience of something? That's still pretty mysterious. So we need to have, get a little more into the anatomy. Again, before we can really dig into the physiology. So let's talk about gray matter versus white matter. So when we're talking about gray matter, particularly in the CNS, this is usually neuron cell bodies. As opposed to white matter, which are myelinated axons. That's why they're white. Myelin is this kind of white fatty stuff. So where you have a lot of myelinated axons, it tends to look whitish. Where you have more just neuron cell bodies. And again, these are kind of more doing kind of the integration, right? These are the things that are actually processing, whereas the myelinated axons are more carrying messages from one place to another. You know, and for completeness, especially if you've been in anatomy, you'll know gray matter can also be unmyelinated axons. There are, like, you know, the little gray commissure in the spinal cord or something. So it's, you know, there is some gray that is unmyelinated axons, but for our class, don't worry about it at all. Let's now kind of lay out where you find the gray matter and the different kinds of white matter in the brain, and then we'll be kind of ready to kind of talk more about function. Okay, so let's say here's somebody's brain. And then we're going to take a coronal section. So when we do that coronal section, again, you have the two hemispheres. So these are my cerebral hemispheres. And actually I can actually do like this even. This little lateral sulcus here is supposed to be this. And so gray matter. The gray matter is found in two main places. It's found in what's called the cerebral cortex. Cortex means bark. So it's a thin layer that's right on the surface. Maybe. So that is the cerebral cortex. It's the gray matter, the neurons that are right on the surface. So like when we are initially kind of talking about different functions of different parts of the brain, and I'm going to be talking about, oh, here in the frontal cortex or the occipital cortex or the temporal cortex or the parietal cortex, we're actually talking about the activity of neurons right on the surface of the brain there. You know, you don't have to dig into the brain if you wanted to activate them. If the skull's off, you could just put an electrode right on the surface of the brain and you'll be activating those neurons or measuring from those neurons, depending on which way. The cortex is doing a lot of the stuff you usually think of the brain as doing. It's like your personality, your impulse control, your sensory processing, your motor control, kind of turning on your muscles to go kick a ball. So a lot of the really important stuff that we'll be talking about is just happening within the cerebral cortex, that thin layer. It's like 5 to 7 neurons thick on the surface of the brain. Gray matter is also found in what are called the deeper nuclei. So there are also like the caudate and the lentiform nucleus and the amygdala, and there's a variety of deeper nuclei that we're going to talk about in more detail as we continue on. So like these are deeper nuclei as opposed to cortex. So deeper nuclei are pockets of neuron cell bodies, gray matter, that are deeper in there. We'll see that a lot of them involved with motor stuff, but also some involved with emotional stuff. Cortex is the thin layer on the surface. All right, so that's gray matter. Now let's talk about the white matter. And again, the white matter is the axons that are interconnecting different parts. So some of what we have are just the little fibers that are interconnecting within a hemisphere. We just call them kind of like association. So these are also called tracts, I should mention. White matter in the CNS are called tracts. So we have association fibers or association tracts. Those are, maybe I'll do them in blue. They're just kind of interconnecting things within a hemisphere. 2, we have commissures or commisural fibers. These connect the right and left sides. So if I did them in yellow, these are the things that are kind of going connecting the right and left hemispheres, so the two hemispheres are talking to each other. The main one, you should know the name of the main commissure that connects. There are a few different commissures, but the one that you should know by name is called, in fact, what is, from anatomy, what is the main commissure connecting the right and left hemispheres?
The corpus callosum.
Exactly, exactly. The main one is called the corpus callosum. There are these fascinating, like, if you've been in psychology, you've probably read about these split brain studies where they cut the corpus callosum. Basically, if someone has really bad epilepsy where one hemisphere is the problem, they'll actually isolate the other hemisphere to kind of save the other hemisphere from further damage. And then you can test these people and see what happens if you cut the connection between the two sides of the brain. And yeah, we'll talk about that a little later. And then finally, 3, we have what are called projection tracts. Maybe I'll do them in green. These are just kind of connecting the lower and higher parts of the brain, right? So projection fibers kind of go up and down. These are the projection fibers. So we have three basic types of white matter. We have the association fibers that stay within a hemisphere to interconnect stuff in here. We have commisural fibers that connect the right and left. And we have projection fibers that connect higher and lower areas of the brain. So that is kind of the extent of the anatomy you need to know for this. So what we can do now is finally kind of lay out what are different parts of the brain doing. And we're going to, if looking at the time for the rest of today, everything we're going to talk about is going to be in the cerebral cortex. So I'm going to be drawing a picture of the brain and the head like this. And we are not going to even be thinking at all about what's inside the brain. Everything that we're going to be talking about is functioning. Is the function of the little neurons right on the surface of the brain, of the cortex, cerebral cortex. All righty then. Let's do this. The brain. For kind of context, here is the head. Where we see that I'm doing the front, your eyes. Most of you have probably known this already from your, if you've taken anatomy, but basically you've got the main lobes are named after the skull bones that they're under. Like, up here, this is all like frontal lobe. I'm going to make a smaller font, it's finer thing, or else you're not going to be able to. The frontal lobe. This one here that's kind of under your temporal bone, this is the temporal lobe. This part here that's under the occipital bone is the occipital lobe. And this part here is the parietal. So what we're going to be doing is talking about what's happening in the cortex in these different regions. Some landmarks, again, a little more anatomy. The division between the parietal and frontal lobe is this sulcus. Remember, sulcus is a little valley. This is called the central sulcus. It's basically a little valley, and it's not that easy to see on a brain, but you can see it. But that separates the parietal and frontal lobes. And it's worth knowing that name because we'll talk about, like on the frontal lobe, there's going to be a gyrus that's right in front of the central sulcus. And we're going to call this the precentral gyrus. There's a gyrus on the backside of the central sulcus. We're going to call that the postcentral gyrus. Right, remember, gyrus is just a ridge. So we have the sulcus, which is the valley, and then these two ridges of brain tissue. So the post-central gyrus, pre-central gyrus are going to be important. We'll talk about them in just a few seconds. There's another gyrus we'll talk about later. All right, Leah, let's move ahead here. All right, let's start with the occipital lobe. Occipital lobe is primarily about vision. So on the very kind of back of the brain there, we have what is called our primary visual cortex. This is the area where you are kind of initially processing visual information. Primary visual cortex gets different names. It's also going to be called V1 because it's the primary visual cortex. Just for completeness, I should also mention it's called Brodmann area 17. The Brodmann numbering scheme just has different numbers that refer to different specific parts of the brain. If you're reading a scientific paper that's studying vision, they'll often they'll use these interchangeably. Primary visual cortex, V1, area 17, are all kind of referring to this part of the brain where visual information is initially being processed. And then, as you can kind of continue further in, we get what are called the kind of visual association areas. So there's primary visual cortex, which is kind of more low-level raw data, but then visual association. We should talk a little bit about, because there's going to be association areas for all of the senses. So like right now, I'm looking at Dana and I see this kind of reddish brownish alteration to the screen. But I don't think like, oh, weird, what's going on? It's like, oh, look, cute, there's a cat, like just hopped on her shoulder, right? You don't look around and see splotches of light and dark in color or this or that. You see objects, you see things, you make sense of the world. That's what when we talk about these visual association areas, it's kind of this more higher level integration of what does this raw data actually mean? I don't want to, this is going to be tricky. Oh, I am going to, shoot, I'm going to, if, what's going on? There we go. Right, if I have, I'm going to have to stop video. Whoop. Hold on. Right here, I have a folder. And you are probably not all freaking out. It's like, what witchcraft is this? That thing was like a big rectangle and now it's just a blue line. That's freaking crazy. And now that the blue line is even half as long as it was, oh my God, and now it's turned back into a big rectangle. It's like, nah, I'm rotating a folder. But that's not what's on your retina. On your retina, there is a short line, there is a longer line, there is a rectangle, right? That's not something that's the raw data. I mean, the raw data is just lines and rectangles, but your experience is, it's an object that's rotating around. It's not freaking me out. You know, it's not like, oh my God, like half the pen disappeared and now half the pen just magically reappeared. It's like, no, it was blocked. And then, you know it was there, and not shocking that it's still there when you take this thing away. But that's not, like, at this point, in fact, like one of the classic so here I'm drawing kind of a classic thing right now. Should have just had it ready to go ahead of time. If I show you this, it looks, most people see it looks kind of like there's like a white triangle sitting on top of 3 black circles, but that's not at all. It's 3 Pac-Men all kind of talking to each other, right? But this is kind of visual association is like, that doesn't make as much sense as there are black circles with a white triangle on top. So your brain is doing a lot of processing with the raw data coming in to kind of make more sense of it, like completing contours. We call these illusory contours because there is no triangle, but you see a triangle. We try to figure out what is actually the underlying reality based on this kind of limited information we're getting. So that's kind of examples of this idea of kind of visual association areas, and they also are very involved with attention stuff, which we'll talk about a bit. But you also have the same kind of idea with somatosensory touch, right? I don't just like say, oh, there is pressure receptor on phalange on like digit blah. It's like it's round and smooth and hard, right? I don't get just the raw data of my touch receptors. I get a bigger thing of kind of what does it feel like? What is the shape and the texture? That's all kind of this association for kind of your somatosensory, the touch stuff. Hearing, you don't just hear frequencies and clicks or whatever. You hear all sorts of sounds, and we'll talk more about hearing. Is even more crazy because every sound is coming in all mixed up together into one vibration on your eardrum, and yet you hear a dog barking in my voice and the dishwasher and someone else yelling at you or whatever. Even though all of those different vibrations are all glommed together into one vibrating eardrum, and yet your brain creates all these auditory objects, just like your visual system creates visual objects. So just kind of putting that out there. The way you process information is you get a lot more than just the raw data. Let's go back to the visual cortex now. So primary visual cortex. This is a good place as any to introduce the idea of mapping, which is something that is pretty interesting in the nervous system. And obviously the word mapping can mean different things. What I mean in this case is this kind of correspondence between the kind of physical world and where things are being processed on the actual cortex. So like in your visual world, if you're looking at something, there's a correspondence between what you're seeing out here and how it's being represented in the neural tissue of your primary visual cortex. So I'm not talking about mapping like vision is here and audition is here and motor is here. I'm talking about within a sense, there is this correspondence between the stimulus and how it's being represented in the cortex that is responding to that sense. So like these are also kind of sad experiments. Like one of the original experiments, they took cats and they showed them a grid. And then while they were showing them the grid, they gave them radioactive glucose. So the neurons that were particularly active are going to take up more glucose because they need more energy. So, and then you sacrifice the cat and you take its primary visual cortex and you put it on a piece of film, and the neurons that had taken up the radioactive glucose are going to expose the film. And so what they found was that there was basically kind of like a grid work on the primary visual cortex in terms of which neurons are responding to the actual view of this grid. So that's what this mapping is. There's this kind of correspondence between the actual stimulus and how it's being represented in the cortex here. The other thing, though, I can mention here that gets really kind of trippy, and this is a way that your brain is not like a computer. So usually people think, okay, here's the kind of raw data, your primary visual cortex, and then it's kind of coming, it gets sent up in here. This is where we have our prefrontal areas or executive areas. These are kind of like your decision making and things like that. These are up in the prefrontal lobes. So there is this thing where you have, axonal tracks that are going from kind of the more raw data to these places where you're going to make decisions about what to do. But what is really kind of tricky is you actually have more tracks that are going the other way. There are actually more axonal tracks that are going backwards and going from these more executive areas to modulate the activity of these primary sensory areas. So for instance, I'm looking on my screen here. Here's, I can't do a good job here. So just on the kind of top of my screen, I have like Dana. This is not a good view. Here's Lindsay. She's smiling. So let's say I am worried that Dana is up to no good. So this is
true.
So this is me. So I am gonna be kind of looking over here. I'm kind of looking at Lindsay here, but I'm kind of my mind, I'm actually kind of paying attention to what is she up to. So we call this like covert attention. Covert attention, I'm looking at Lindsay, but really in my mind, I'm kind of focusing on what's happening out on the side of my visual field where Dana is. But Dana doesn't actually have it's covert because it looks like I'm paying attention to Lindsay, when in fact, I'm paying attention to Dana. So it turns out when you do this, it actually changes how this mapping, how much of your primary visual cortex is being allocated to process information about the area of space where Dana is as opposed to where Lindsay is. So if we kind of go back over to here, this activity here in these executive areas and these kind of decision-making areas is now going back and actually kind of remapping the allocation of your neural resources here. So you've got, I would have more of my visual cortex dedicated to actually what exactly is Dana doing over there? As opposed to if I wasn't, I would have had my primary visual cortex focusing more on what I'm looking straight at, which would be Lindsay there. So does that make sense? So it's kind of a fascinating thing, like this idea of attention really changes just the way you actually process your world around you. It's not just like, it's not like just all the information comes in and then you get to sift through it. It's like how you actually see the world kind of is influenced by how you're looking at it, it's just kind of trippy, which is useful to remember too, I think. So, all right. So visual stuff. Let's continue now. All right, so visual, they're in the occipital lobe. Let's do somatosensory. Actually, let's do this. Let's do here in the temporal lobe. This is where we're going to have our primary auditory cortex. So this is where you are processing sound. There are also going to be association areas as you kind of go out from there. And there's mapping here as well, except the mapping, instead of being about visual space being represented in the cortex, it's going to be about frequency of the sound being represented in the cortex. So here we're going to have mapping as well, but we're going to have what's called tonotopy. So depending on which frequencies, you're going to be on one part versus another part of another part of this cortex here. So that's another version of mapping. Like different parts, different frequencies are kind of represented in a kind of regular way across the auditory cortex. And there's some other stuff on the temporal lobe, but let's leave it there for now. Let's go to so right here, this is what we call the post-central gyrus. So you've met the post-central gyrus before. It's just that ridge in the parietal lobe that's just behind that central sulcus. This is what is going to be our primary somatosensory cortex. So somatosensory, those are like the body senses. These are the general senses. This is like touch and stuff all around your body. You know, and again, there's going to be association areas that are associated with that, right? Because you don't just feel just raw data for touch. Like I said, it's round and it's hard and it's smooth or whatever. It's wet, right? There's all these different things that you get from both the touch and the temperature and things where you can get a more complete sense of what actually is this thing that you're touching or touching you. And there is mapping here as well, but we'll do that as soon as we do the motor cortex. Over here, this precentral gyrus this is our primary motor cortex. So this is what is sending messages out to your skeletal muscle to move your, remember somatic, you want to kick a ball or move your arm or this or that. We're going to see there's a variety of other parts of the brain involved with motor as well. Some of these deeper nuclei, the basal nuclei and stuff. But there was this guy named Penfield back in the 50s that did these famous experiments with these epilepsy patients where he would take an electrode and touch along this precentral gyrus, and you could make different parts of the body twitch. That's kind of how they started realizing that this is involved with motor control. Same thing over in the postcentral gyrus. As he stimulated little parts of the cortex along the postcentral gyrus, people felt things on different parts of their body. So precentral gyrus, and for a long time, they thought it was kind of really just more like activating a muscle. Now it seems actually a little more complicated than that, but for our purposes, let's just say it's kind of more direct control of your muscles. There is what we call the motor association area or premotor area. Right, because when you use your muscles, you don't think about your muscles. You think about what you want to do. It's like, I want to scratch my nose, right? The reality is I had to bend my arm. That was like probably biceps brachii and brachialis. I had to actually adduct my arm, which was probably pec major. I have to actually bend my fingers, which is these like flexor digitorum, like, superficialis and profundus. I've got all this stuff going on, but I'm not thinking about, even though I know the muscles because I teach anatomy, I'm not thinking about the muscles. I'm thinking, I'm going to scratch my nose. So this premotor, motor association seems to be part of that planning out, like you know what you want to do and kind of do the planning so the low level stuff happens without you having to deal with it. You don't have to think about which muscles are you moving. You just have to think about what you want your body to do. Later on, we're going to talk about kind of closing the loop, like how your body knows what it did, or if it's actually completed the task and you're done. There's going to be a lot of feedback involved here, and we're going to have the cerebellum involved here as well to make sure that your motor movements are accurate and complete and all that. But for right now, let's just kind of introduce this part of the brain here. Both the somatosensory cortex and the primary motor cortex have what are called somatotopy. So this is also a type of mapping. Soma means body, so it's mapping how the body is represented or corresponds to the different parts of these different cortices. And again, they found this out just as they stimulated the little parts of the cortex with an electrode to see which part of the body twitched in the primary motor cortex, or which part of the body felt a little twinge on the somatosensory cortex. And they could just map out that correspondence between where on the body am I and where in the cortex is that part of the body represented. And let me go. I can just show, this makes more sense if I just show it on a screen. So the classic thing here is, let's do images back. We're going to, wait, what are we going to do? Let's do somatotopy. Here we go. So this here is showing somatotopy. It's showing like, here is the hand, the thumb, the index finger. In fact, let's see if we can view this bigger. There we go. So this is supposed to be the hemisphere, right? This is the cortex, is this darker part. And it's just showing that the different parts of the body are represented on different parts of the cortex. And you have a similar kind of layout, both for the motor and for the somatosensory. So here you can see motor and sensory are kind of similar. You can also see that it is not kind of proportional just to the size of what parts of the body have more than their fair share of representation in this mapping.
The face and hands.
Yeah, the face and the hands. So your hands, which are both incredibly dexterous, can do really fine motor stuff, and also incredibly good at just interpreting, like blind people can like just read little dots by zipping along, just using the information coming in their fingertips. Your face and your mouth is like critical for both making speech and for expressions and all of that. So you have like in general, almost like a third of the cortex is your face and tongue and mouth. A third is your hands, and then everything else gets the other third. There's a word people use, homunculus, it means little man, to kind of describe what would you kind of look like. You know, here, these, I love these. You know, this is like if somebody's body proportions were the same as how they're represented on the cortex. You know, you get like things are big hands, big face, mouth, and tongue and stuff. So anyway, homunculus. You know, and there is some plasticity. They've done these kind of, again, really kind of interesting, kind of mean experiments. Well, they'll like teach a monkey to do different tasks with its hands, and they'll map out, they'll have access to its brain, and they'll see how is their hand represented in the cortex. And then they'll cut off one of the fingers. So now all of a sudden there's no longer anything that corresponds to that. They think some of this is part of what phantom pain is about. Sometimes when people lose a limb, they can still feel it. And sometimes they can even feel excruciating pain in a limb that actually doesn't exist anymore. And they think that that comes from that part of the brain that represented that limb is still there. And if there's surrounding activity that somehow bleeds over and stimulates activity in that, you can actually have this experience like, oh, there's some electrical activity, some neural activity from this part of the brain representing this limb, even though that limb doesn't exist anymore, you can have the experience that is there. So it can be kind of freaky, or it can actually be pretty debilitating if it's painful experience. But they also did find that when they trained the monkeys to keep doing the tasks with the amputated finger gone, that there was actually some remapping, that the parts of the cortex that are for the remaining fingers kind of started encroaching in on that area. So that's another concept I should mention, and then we'll, right before we do our break, this idea of plasticity. Plasticity is the potential for the brain to actually kind of change what it's doing. Like having one part of the brain kind of remap and start doing something a little different. In general, there is some capacity for plasticity. It's much higher in little kids. Little kids' brains, you can take a big chunk out of them and the remaining parts will actually reconfigure to probably take over the missing functions. As you get older, if you take a big chunk out of your visual cortex, you're just going to not have vision in that part of your visual field anymore. But there is some ability for plasticity. People who have strokes and lose function because part of the brain tissue died, over months even, you can start having recovery of function as the brain kind of rewires and remaps. When I first started teaching this class, the dogma was that, you're born with all the neurons in your brain that you're going to have, and you never get any more. And now we know that's not true. There are kind of neuron stem cells in the hippocampus that can then migrate out to other parts of the brain and reconnect and rewire things. So there is more of a capacity for this plasticity than we thought many years ago, but it's still limited. Like if you have, some traumatic brain injury, either through like a stroke or through some physical trauma, there's a good chance that you will have kind of long-lasting deficits. But there is also the possibility at least some of those deficits will get better or resolve over time, depending. So we'll talk more about this as well as we go on. I think I might have mentioned neuroplasticity. One of the trippier things that like one of the studies of a couple of years ago is plasticity is related to physical activity. Like people who had like problems with their processing of visual information. Like one of their eyes wasn't working so well as they were growing up, and that part of the cortex didn't develop so well. And so you're trying to do these tasks now as you're older, seeing can we rewire the brain responding to the visual information and kind of make it work properly. What they found was if people just did this task looking at images and things, it didn't make a difference. It really didn't seem to help much. But if they did the same task while they were actually on a stationary bike, it really did make a difference. Like there was something about these like neurotrophic factors, these things that actually affect the growth of brain cells that were more active, and all, the brain was more able to adapt and rewire and heal when people were active. And there are actual molecules developed. And there's actually stuff now just in the last couple of months in Science magazine that exercise releases stuff that's released by the liver that actually helps reduce cognitive decline and aging. So exercise really has pretty measurable effects on your brain being able to like adapt to things, as well as not get kind of as much degenerative damage as you get older. So I would say there's a lot of evidence, measurable evidence that exercise is good for you for lots of reasons, which I think is particularly worth like reiterating right now during COVID, when it's like really easy to just be a blob in front of a Zoom stuff. So let us continue a little bit with the cerebral cortex. All right, so talked about visual in the occipital, somatosensory here in the parietal in this post-central gyrus, motor here in the precentral gyrus. And I should mention the little mnemonic, like the way I would always make sure I remembered which was motor and which one was somatosensory was alphabetically, M is in front of S. So motor is the pre-central gyrus. It's the one that's in the frontal lobe. Sensory or somatosensory is comes after M in the alphabet, and it's behind. It's in the post-central gyrus there on the parietal lobe. We talked about the auditory here. This area here, this what we call prefrontal area. So prefrontal is interesting. Like, if you have damage to your occipital, you're going to have trouble with vision. If you have some stroke that damages somatosensory, you're not going to feel stuff on your body. If you have stuff that damages the motor, you're going to be paralyzed. If you have damage to the prefrontal area, the deficits are not quite similar. The deficits are more around your personality. So prefrontal, this is what we call kind of like executive functions. This is like decision making. Right? The executive is the one who figures out, how should we allocate our resources? How are we going to respond to this particular thing? What are we going to do next? What's the future looking like? Impulse control. When I was in school, we would have, we'd get to meet patients with different kinds of neurological disorders. And there was this one guy who had like head planted without a helmet at high speed from a motorcycle accident and had kind of messed up his prefrontal areas. And, one of the things they talked about with him was, he would just see some woman he was attracted to and just reach out and try to grab her breasts. There was like no there was no like piece in between, like, oh, I'm attracted and what's appropriate. It was just like, this impulse control was not working properly. The classic Phineas Gage guy who had like a steel rod go through his head and messed up his prefrontal area, supposedly just totally changed his personality. He went from being kind of a nice, sweet guy to being a jerk. So, prefrontal, executive functions, decision-making, impulse control, probably imagination, whatever. But it's not about the more kind of physical things. Sometimes they call this like orbitofrontal cortex, because orbit is like where your eye is. So it's like kind of the part of the frontal lobe that's closer to like where your eyeballs are there. The orbitofrontal cortex. But but it's like kind of where you decide what to do with things. So, and I should mention, one of the fascinating things that's come out of some of the research looking at the development of the brain with kind of the more modern, MRI techniques, you can actually kind of look at white matter and watch how does the white matter and gray matter change from being a little kid to being an adult. And what they've found is that the wiring up of the brain, like when you're born, you've got like twice as many neurons as you have as an adult. And through experience and stuff, the neural circuits kind of wire up and get refined. And as the circuits become more refined, you get more white matter, more of the wiring kind of connecting things up in their final ways. And what you can see in these studies where they look at white matter through like adolescence to adulthood is that the wiring, like if this is the brain, that the wiring seems to kind of start more from the back and go up to the front, and that the implications are that the complete wiring of the prefrontal areas isn't done until your late teens, early 20s. So there's still kind of development and kind of circuitry kind of resolving up until you're already like past your teens into your early 20s. And with the implications that since this is the area that's involved with like impulse control and decision making, that those parts of your brain are not totally online until like you're out of high school. So the fact that like a high school kid is being impulsive and doing stupid things isn't necessarily so much of a character flaw as partly like the hardware is still coming online there. So it's it brings up interesting questions around like accountability and things like that even. So anyway, just I always thought that's kind of interesting to think about. The other thing I will mention, let's just do the language stuff and then we'll leave it there. There's more stuff for the cerebrum and even the cortex that we can talk about, but let me talk about language and then we'll move into the lab. So actually to talk about language, we need to talk about laterality of the two hemispheres. So obviously I'm drawing this brain from the side here. You're looking, if this is this side of his head, that's like the left side of his head. But obviously there are two hemispheres, right? Like if you're actually looking at them. There's the left hemisphere and the right hemisphere. And we have this idea of laterality. How the two sides are different from each other. And they're very similar, but they're not exactly the same. One thing that is interesting about the laterality is, for the most part, the two sides of the brain are cross-wired to the opposite sides of the body. So that's really important to know about, especially if someone has a stroke or something that damages. Like if you damage your motor cortex on the right side of your brain, where are the deficits going to show up?
The left?
The left. Yeah, you're going to get paralysis on the left side of your body. So it's important to kind of realize that anything that affects one side of the brain is actually going to show up with symptoms on the opposite side of the body. So, you know, there's this word in anatomy, we say decussation, right? All the axons tend to decussate. They cross over from one side of the body to the other as they are leaving the brain to connect up to whatever part of the body they connect to. So it's actually physically the axons cross over from one side of the brain to the opposite side of the body. So it's not like a magic thing. It's the physical wiring crosses over. So that's one thing. Other things, you know, right and left are different in like language. Language is on left for like 90% of people. So if we're going to talk about like Broca's area, Wernicke's area, these areas that are primarily for processing and production of language, for like 90% of the population, language is done on the left side of the brain. That means, you know, right side of the brain, maybe 5%. These people who have language on the opposite side tend to be lefties. They're left-handed people. So for whatever reason, when language, whatever side of your brain is dealing with language, the other side is usually your dominant hand. And then it's maybe mixed, 5%. It's mainly on the left. If you had to make a guess for somebody, but they actually will do these experiments, like if they are going to go in and do brain surgery on you, right? And they actually care a lot about which side of the brain is the language on, right? If you go in and you mess something up a little bit in the motor areas, and maybe somebody's a little less coordinated with their hand or something, it's like, whatever, I can live with that. But if you all of a sudden lose the ability to speak, that's catastrophic. So before they actually go in and do brain surgery that might get into any of these areas, they want to know which side of the brain on this particular patient is language. And so they actually have this test where they'll, you have the carotid arteries going up. They can actually put a anesthetic in one carotid artery versus the other carotid artery while someone's awake and have them kind of yapping. And when you hit the one that feeds the hemisphere that is responsible for language for that person, they'll just go mute. So like, one of my friends sat in on one of these things. He says just kind of the person said that. And you stop talking if you anesthetize the hemisphere that is responsible for language. So you can actually do that relatively non-invasively, although you gotta stick chemicals up into their arteries. So language. And I should also mention there is some of that whole thing, right brain, left brain, that does come from some of the realities of, the left brain does tend to be more analytical, taking kind of the more mathematical information out of things, versus like one example, like for the language, like when I'm saying language is on the left, I mean more the semantic content of the language, like what do the words actually mean, the definitions and the grammar, right? And the right side, though, is getting more of the affect, of the emotional content, right? And a lot of the meaning isn't just what the words mean. It has to do with the actual intonation and, like examples I like to use if, I could, kind of going back now I have like Gene up at the top here, so I can say like, depending on my expression and my tone of voice, the exact same words, even if the semantic content, like, hey, Gene, it's nice to see you in the Zoom meeting today. It's just, that means one thing. And if I like, oh, Gene, nice to see you in the Zoom meeting today, it's really different, even though the words mean the exact same thing. What it actually means at a bigger level is very different. So, there you have like kind of right brain, left brain, like one part getting more of the analytical, what does these words mean? And another side getting more into kind of the music of the language. So just kind of putting that out there. There's lots of different things that people look at, but that right brain, left brain thing is kind of fascinating. That even though both sides of the brain, both do sound, both do vision, both do this and that, they often kind of take slightly different aspects of the information. And again, the left side tends to be a little more of the analytical side versus the kind of, poetic side. What else? So let's take a look at the two main parts that people tend to associate with language. So kind of in this kind of premotor area down below, this is what they call Broca's area. This is production of language. Right, it's actually, if you actually stop and think about it and put some attention on it, it's actually kind of bizarre and magical how talking works, right? I have, like, whenever I start talking about it, it gets weird for me. It's like, I just know what I want to say, and somehow my mouth and my tongue and everything just goes yap, 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 blue, and the words just spew out of my mouth. But it's really freaking complicated. Like somehow these ideas of words become these motor programs, become vibrations that now go across the room and tickle your eardrums and you hear my voice. So Broca's area is that part that is kind of making that connection between what do you want to say and the actual physical manipulation of your motor system to make the words actually happen. When people have damage to this, so we should, the word aphasia is just a condition where you have a problem with language. So if you have damage, like what we call Broca's aphasia, this would be another word, you could call this expressive aphasia. You have problem expressing what you want to say. Or sometimes they call it non-fluent aphasia. Meaning you're not fluent, you can't make the words come out. So that is one kind of aphasia that comes from this damage to the area that makes that produces the language. There's another area kind of more around here called Wernicke's area. And they get their name based on these neurologists from the 1800s who had patients. Like Paul Broca was this neurologist in the 1800s. He had this famous patient, like patient Tan, because all patient Tan could do was go Tan, 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 Tan. And after he died, they did an autopsy and found, oh, he had a lesion right around here. That was kind of like the beginning of understanding that. Wernicke was from another dude named Wernicke who had a patient who couldn't understand This is about kind of understanding of language. And I should mention language is more complicated than just these two, but this is like a good place to kind of get into it. So when people have Wernicke's aphasia, the problem isn't making words, it's understanding what words mean. So sometimes they call it receptive aphasia. Because you are not receiving the information. You can't let it in, figure out what the meaning is. Sometimes it's called fluent aphasia. Because people can talk, they can make words. And they do make words often, but the words don't mean anything. Right? So there's no meaning behind the words. I'll show you a video in just a few moments. So these are like the two, and a lot of times aphasias are mixed. Like the main arteries that go into one of these goes into the other. So sometimes you get damage to both of them. And then it can actually make diagnoses really tricky. It's like, does this person know what they want to say and they just can't say it? Or they don't know what they want to say, and that's part of why they aren't saying it. Or maybe they don't know what they want to say and they couldn't even if they did know. Or so, diagnosing these mixed aphasias can get really tricky. And I realize I've been using the word strokes and stuff. I should just define that really briefly, and then I'll show you some examples of these, and then we will move on to the lab. Stroke. So we're not talking about that. So we're going to say CVA, Cerebrovascular Accident. So cerebrovascular accident is when something goes wrong that impairs the blood flow to part of the brain tissue. It can happen because there's a blockage of the blood vessel, like some clot broke free and then got stuck in the vessel and created kind of downstream ischemia, so no more blood. So ischemic stroke where there's no blood flow. You can have hemorrhagic strokes where a blood vessel bursts. So then you just get bleeding in the brain, and obviously the blood isn't continuing on to where it's supposed to get to. So there's different versions of strokes, CVAs. Obviously, this is going to be bad. So the cells that are not getting adequate blood are going to start dying. Sometimes you get what's called a TIA, transient ischemic attack. So ischemia is just lack of blood flow. Transient meaning it's happening, but then it resolves. So this is like where somebody is like, whoa, everything's getting weird and half my body stopped working, but now it's working again. Right? So this is obviously not as worrisome as something that just keeps going and the cells start dying off. That being said, even if things resolve, sometimes there is some residual damage, like the little mini strokes. You can actually start ultimately getting kind of more long-term kind of dementia or things like that happening when people, even if each one is not that worrisome, like some major stroke, you still want to, they're not good. Like, do people know like different, like some of the ways people talk about, like if somebody's getting weird, how you might be able to tell in the moment that something's going wrong with the blood flow to their brain?
I think of when like one side of their face kind of droops.
Yeah, so that's a big thing. Or if you even have them like stick out their tongue, and if the tongue kind of goes off to one side when they're trying to stick it straight out, that's a sign that, you know, one side of their musculature isn't operating. So yeah, if somebody is having a stroke, the sooner you can find that out, the better chance, because there are drugs we'll talk about when we do hemostasis, we'll talk about TPA, which, you know, just Genentech drug that, you know, the clot buster drug. Like if you can get somebody to the hospital within an hour, there's a good chance they're going to be minimal damage. Whereas if you don't get them to the hospital, there could be like more devastating consequences. So just kind of, so strokes happen a bunch, and not just old people. I actually, at this point, you know, by the time I was in my 40s, I had two friends who had pretty massive strokes, one person like never recovered, one person slowly recovering and it's never going to be as mobile as they were. So it's definitely, they're kind of scary. And that whole COVID-19 thing seems much more likely to get a stroke if you got COVID than not. So another reason to avoid getting COVID-19. All right, but back to Wernicke's aphasia versus Broca's aphasia. Again, Wernicke, receptive aphasia, you can't actually understand the words, but you still have the ability to make them. As opposed to Broca's, you understand, but you don't have the ability to actually convert the idea of what you want to say into an actual word. So I'll show you a couple of videos. There's lots more, but these are a couple that I found online that I think, you know, illustrate this stuff very well. So let's go. I'm going to share. So these are just sitting here in the module for today here. Aphasia examples. Oh, I'm going to publish. So there it is. Aphasia examples. So these are just here. You can look at them later. Here, this is non-fluent aphasia. So here's this girl who had a stroke and she's talking about it.
Okay. So what's your name?
Scott. Oh, no. Sarah Scott.
That's right. And how old are you?
I can't.
Try.
I can't.
You're 19. 19. Yeah. And what happened to you?
Stroke.
You had a stroke last year.
Yes.
And what happened? Can you remember what happened?
School and English class.
Okay.
And I book and I read it aloud. But I can't because stroke. And so I stand there and also it's the same as the same kind of thing as you know,
pins and needles.
yeah,
like
the same, and also as well.
All right, so you get the sense here that she really has a lot of, and it's not affecting cognitive at all, right? She totally knows what she wants to say. She's totally just as sharp as she ever was, but she just is struggling so hard to like make the words happen, as opposed to the other kind, like a receptive aphasia, where somebody has the ability to make sounds, but is not really making sense of what words mean.
Hi, Byron. How are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talked with the people for them over there. They're diving for them at the moment. But they'll save in the moment. He'll have water very soon for him. With luck for him.
So we're on a cruise and we're about to
We
get to
will store it
June.
right here and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment, they don't show a darn thing.
The iPad that we were doing. We, like here?
I'd like my change for me and change hands for me. It would happen. I would talk with Donna sometimes. We're out with them. Other people are working with them. I'm very happy with them.
Good.
This girl was very good and happy. And I play golf and hit other trees. We play out with the hands. We save a lot of hands on hold for us, other hands. I don't know what you get, but I talk with a lot of hand for him. Sometime. Am I talk of any more to say it?
All right. Thank you very much.
Thank you very much. I appreciate it. And I hope the world lasts for you.
Thank you. It's been a pleasure. Bye-bye.
Have a good day.
All right. So there you get a sense, like the guy has no trouble yapping, but it's just what they call word salad, and it gets trickier even to know, like, does he even know what she's asking him? It's like it's really.
Can you tell us your name?
Mike Caputo.
So anyway, so just kind of give you a sense, and because these aphasias are very common results of strokes, and strokes are pretty common. So this is definitely something to be aware of. All right, so now it's time to go back to lab.